Respiration in Plants, Part 1. To live, all of us breathe. But to live, why is it so essential to breathe? When we breathe, what happens? Also, including plants and microbes, do all living organisms breathe? If so, how? For carrying out daily life activities, all living organisms need energy, be it transport, absorption, movement, reproduction, or even breathing. From where does all this energy come? For energy, we know we eat food, but from food, how is this energy taken? How is this energy utilized? Is the same amount of energy given by all foods? Do plants eat? From where do plants get their energy? Do microorganisms eat food for their energy requirements? You may wonder, they may seem to be very disconnected at the several questions raised above, but in reality the process of release of energy from food is very much connected to the process of breathing. Let us try and understand how this happens. By oxidation of some macromolecules that we call food, all the energy required for life processes is obtained. Their own food is only prepared by green plants and cyanobacteria. They trap light energy and convert it into chemical energy by the process of photosynthesis, and that is stored in the bonds of carbohydrates like glucose, sucrose, and starch. We must remember that not all cells, tissues, and organs in green plants photosynthesize, but photosynthesis is carried out only in cells containing chloroplasts that are most often located in the superficial layers. Hence, even all other organs, tissues, and cells that are non-green in green plants need food for oxidation. Hence, to all non-green parts, food has to be translocated. Animals are heterotrophic, that is, directly or indirectly, they obtain their food from plants. Fungi, like saprophytes, are dependent on dead and decaying matter. What is important to recognize is that for life processes, all the food that is required ultimately comes from photosynthesis. This chapter deals with the mechanism of breakdown of food materials within the cell to release energy or cellular respiration and the trapping of this energy for ATP synthesis. Of course, within the chloroplast, photosynthesis takes place, whereas in the cytoplasm and in the mitochondria, the breakdown of complex molecules to yield energy takes place. Through oxidation, breaking of the carbon-carbon bond of complex compounds within the cells leading to the release of a considerable amount of energy is called respiration. During this process, the compounds that are oxidized are known as respiratory substrates. Carbohydrates usually are oxidized to release energy, but under certain conditions in some plants, proteins, fats, and even organic acids can be used as respiratory substrates. Within a cell, during oxidation, all the energy contained in respiratory substrates is not released free into the cell or in a single step. It is released in a series of slow stepwise reactions controlled by enzymes, and in the form of ATP, it is trapped as chemical energy. Hence, in respiration, the energy released by oxidation is not, or rather cannot be used directly, but is used to synthesize ATP, which is broken down whenever and wherever energy needs to be utilized. Hence, ATP acts as the energy currency of the cell. In ATP, this energy trapped is utilized in various energy requiring processes of the organisms, and during respiration in the cell, the carbon skeleton produced is used as precursors for biosynthesis of other molecules. Do plants breathe? Well, the answer to this question is not quite so direct. Yes, for respiration to occur, plants require oxygen and they also give out carbon dioxide. Hence, to ensure the availability of oxygen, plants have systems in place. Plants, unlike animals, have stomata and lenticels for gaseous exchange and have no specialized organs for this purpose. There are several reasons as to why, without respiratory organs, the plants can get along. First, gas exchange needs is taken care of by each plant on its own. From one plant part to another, there is very little transport of gases. Second, for gas exchange, plants do not present great demands. At rates far lower than animals do root, stem, and leaves respire. Large volumes of gases are exchanged only during photosynthesis, and during these periods each leaf is well adapted to take care of its own needs. The availability of oxygen in these cells is not a problem when they photosynthesize, since within the cell oxygen is released. Third, even in large bulky plants, the distance that gases must diffuse is not great. In a plant quite close to the surface, each living cell is located. You may ask, this is true for leaves, but what about thick woody stem and root? In stems, inside and beneath the bark, the living cells are organized in thin layers. They also have openings called lenticels. In the interior, the cells provide only mechanical support as they are dead. Thus, in a plant, most cells have at least a part of their surface in contact with air. In leaves, stems, and roots, this is also facilitated by the loose packing of parenchyma cells, which provide an interconnected network of air spaces. A complete combustion of glucose, which produces end products such as carbon dioxide and water, and yields energy, most of which is given out as heat to the cell. If this energy is to be useful, it should be able to utilize it to synthesize other molecules that the cell requires. To catabolize the glucose molecule, the plant cell uses the strategy in such a way that not all the liberated energy goes out as heat. The key is to oxidize glucose in several small steps, but not in one step, and enabling some steps to be just large enough such that the energy released can be coupled to ATP synthesis. Oxygen is utilized during the process of respiration, and as products, water, carbon dioxide, and energy are released. Oxygen is required for combustion reaction. But some cells live where oxygen may or may not be available. Where oxygen is not available, can you think of such organisms and situations? There are sufficient reasons to believe that in an atmosphere that lacked oxygen, the first cells on this planet lived. We know of several living organisms, even among present day, that are adapted to anaerobic conditions. Some of these organisms are facultative anaerobes, while the requirement for anaerobic condition is obligate in others. All living organisms in any case retain the enzymatic machinery without the help of oxygen to partially oxidize glucose. This breakdown of glucose to pyruvic acid is called glycolysis. Glycolysis. From the Greek words, the term glycolysis has originated glycose for sugar and lysis for splitting. Gustav Embden, Otto Meyerhoff, and J. Pranas have given the scheme of glycolysis, and often it is referred to as the EMP pathway. It is the only process in respiration in anaerobic organisms. In the cytoplasm of the cell, glycolysis occurs and it is present in all living organisms. In this process, two molecules of pyruvic acid are formed by the partial oxidation of glucose. This glucose is derived from sucrose in plants, which is the end product of photosynthesis, or from storage carbohydrates. By the enzyme invertase, sucrose is converted into glucose and fructose, and these two monosaccharides readily enter the glycolytic pathway. Glucose 6-phosphate is formed when glucose and fructose are phosphorylated by the activity of the enzyme hexokinase. The glucose that is phosphorylated then isomerizes to produce fructose 6-phosphate. Subsequent steps are the same of metabolism of glucose and fructose. In glycolysis, under the control of different enzymes, a chain of 10 reactions takes place to produce pyruvate and from glucose. Please note while studying the steps of glycolysis at which utilization or synthesis of ATP or NADH plus hydrogen ion takes place. ATP is utilized at two steps. First, when glucose is converted to glucose 6-phosphate, and second, when fructose 6-phosphate is converted to fructose 1,6 diphosphate. Fructose 1,6 diphosphate splits into dihydroxyacetone phosphate and 3-phosphoglyceraldehyde or PGAL. In one of the steps, we find that NADH plus hydrogen ion is formed from NAD ion. This takes place in the conversion of 3-phosphoglyceraldehyde or PGAL to 1,3-bisphosphoglycerate DPGA. From PGAL in the form of two hydrogen atoms, two redox equivalents are removed and transferred to a molecule of NAD ion. PGAL is oxidized and the inorganic phosphate is to get converted to DPGA. It is noted that the conversion of DPGA to 3-phosphoglyceric acid or PGA is also an energy yielding process, and this energy is trapped by the formation of ATP. During the conversion of PEP to pyruvic acid, another ATP is synthesized. Can you then calculate in this pathway how many ATP molecules from one glucose molecule are directly synthesized? Then the key product of glycolysis is pyruvic acid. What is the metabolic fate of pyruvate? This depends on the need of cells. Different cells handle pyruvic acid produced by glycolysis in three major ways. They are aerobic respiration, lactic acid fermentation, and alcoholic fermentation. In many prokaryotes and unicellular eukaryotes, under an aerobic condition, fermentation takes place. Organisms adopt Krebs cycle for the complete oxidation of glucose to carbon dioxide and water, which is also called aerobic respiration, and this requires oxygen supply. Fermentation. The incomplete oxidation of glucose in fermentation by yeast is achieved under an aerobic condition where by sets of reactions pyruvic acid is converted to carbon dioxide and ethanol. These reactions are catalyzed by the enzymes pyruvic acid decarboxylase and alcohol dehydrogenase. Other organisms such as some bacteria from pyruvic acid produce lactic acid. In animal cells also, like the muscles during exercise for cellular respiration, when oxygen is inadequate, pyruvic acid is reduced by lactate dehydrogenase to lactic acid. In both the processes, the reducing agent is NADH and hydrogen ion, which is reoxidized to NAD ion. Not much energy is released in both lactic acid and alcohol fermentation. In glucose, less than 7% of the energy is released and not all of it is trapped as high energy bonds of ATP. Also, the processes are hazardous and either alcohol or acid is produced. When one molecule of glucose is fermented to alcohol or lactic acid, what is the net ATPs that is synthesized? When the concentration of alcohol reaches to about 13%, yeast poisons themselves to death. In beverages, what would be the maximum concentration of alcohol that are naturally fermented? How do you think alcoholic beverages of alcohol content greater than this concentration are obtained? What then is the process by which complete oxidation of glucose can the organisms carry out and extract the stored energy to synthesize a large number of ATP molecules which are required for cellular metabolism? These steps in eukaryotes require oxygen, and this takes place within the mitochondria. Aerobic respiration in the presence of oxygen leads to the complete oxidation of organic substances and releases carbon dioxide, water, and a large amount of energy that is present in the substrate. In higher organisms, this type of respiration is most common. Thank you so much for watching. Please share this video with your friends. Leave your comments below and please subscribe to my channel for more content. Click that bell notification that would let you know when a new video is uploaded. Thank you so much for watching. Once again, please subscribe to my channel for more content. For more videos, please check the description box down below.